Good evening, everyone. Very welcome in this special evening where we will introduce this year's Nobel Prizes by now almost famous Nobel Lectures of the University of Twente. This is a co-production of the Studium Generale and the Faculty of Science and Technology, as well as the study associations Argo, Alembic and Paradox. So very welcome to everyone, but maybe most welcome to all the students that are here in the audience. As you see here on this list, indeed, it's already quite a tradition. We started in 2007, discussing in these meetings the recently awarded Nobel Prizes by experts in the particular fields, preferably people that also had personal interactions with the new Nobel laureates, and it's quite a challenge always to find the right people. But this year we have two experts from our own university who can introduce the Nobel Prize for Chemistry and for Physics, and as a special treat also the Nobel Prize for Medicine and Physiology, as will be shown today. So these speakers are Professor Jurriaan Huskens and Professor Pepijn Pinkse. I will introduce them in a minute. I would also like to thank Peter Timmerman and the crew of the Studium Generale, as well as Carinza Broersen and Gert-Jan Koster, who have helped also in trying to, on a very short notice, trying to find the right experts for the Nobel Prizes for this year. I think we're in for a very interesting program. Of course, there's also the possibility to discuss, so if you have questions after the introductions by the experts, we'll have time to discuss the various prizes and the ins and outs of their discoveries. So the first prize that will be introduced today is the Nobel Prize for Chemistry. Please, Jurriaan, if you can come up. The prize will be introduced by Professor Jurriaan Huskens, who is a professor in chemical engineering here of the University of Twente. He's the group leader of the group on molecular nanofabrication, so he's a real nanotechnology and chemistry expert. He studied at the Technical University of Eindhoven and did his PhD at the Technical University of Delft. So it's a kind of three TU person, but he was a postdoc then afterwards in Texas, University of Texas in Dallas. After that, he went to the Max Planck Institute for Kohlenforschung in Mülheim in Germany. And since 1989, he's here at the University of Twente. In between, afterwards, he also had a few visits, short stays, at the Northwestern University in Chicago and at Durham University. And he's also winner of various awards, including the MBO Fiji Prize in 2008, if I remember well. Please, Jurriaan, tell us about the click chemistry.
Yes, very good. First of all, welcome to all of you. So this year's prize was awarded for click chemistry, and so I would like to know who of you is involved with chemistry. Who's studying chemistry here? Oh, I studied. I see a few hands. Yeah. Who thinks chemistry is, of course, the most fun? Good. Excellent. So, because chemistry is a special science, I would say. So, slides are up. So I'm going to show you a bit of what the Nobel Prizes, of course, is about, and who won it this year. And here is the picture with already cartoons of the three winners that you probably might have seen in an announcement for this click chemistry. And one of the quotes that was cited there, it just says click. Well, actually, you can put your ear near a reaction tube, but it doesn't do that. It doesn't make any sounds, but the click chemistry comes from, I will explain in a minute. It has to do with the ease of which things go. And that's, I think, depicted nicely in the image. So it's like buckling up, so to say. So you make a bond between molecules that makes it into a larger molecule. And that's basically what the whole prize is all about. So for the non-chemist or for the people who have had in the distant past maybe a little bit of chemistry, this is a complicated chemical molecule, the structure of it. It's Taxol. Taxol is an anti-cancer drug, And it's also a drug, it's a compound that is being made by plants. Taxus trees makes this molecule. And actually you can get it from the leaves if you cut the taxol plant. And people have been fascinated for decades on how to make such molecules. And that's often what chemistry is associated with, how to make molecules and how to make new molecules. But also existing ones, natural molecules like this one. So there are whole groups and people devoting their lives in designing synthetic methods in order to make complicated molecules and learn how to do that in the best possible ways. And with Taxol, I will not go into detail, but not so long ago, in 2021, there was finally a chemical route developed on how to make Taxol. And you see here part of the synthetic process. So this is a scheme from that particular paper. And I will not go through one by one, yeah, so let's not do that today. But if you read stuff, you see all kinds of reactions depicted, solvents being used, all kinds of organic, smelly solvents, volatile, flammable solvents, lots of waste coming out. You see in the beginning here, the simplest compounds are being used on 20-gram scale, and what at the end comes out is a few milligrams of Taxol. So yes, we can make it. Can we make it very efficiently? So. So chemistry is hard. That's the top subtitle that I gave this slide. And what are the problems? It's actually that CC bonds are difficult to make. So big molecules, carbon is often the atom of choice. So that's what is present in practically all molecules around. Definitely the part that we call organic molecules, and therefore also all the molecules that are present in our body and in living systems in general, they all contain carbon. And they're based on carbon chemistry. And where nature has learned how to build complicated molecules well and in efficient ways, this is still a challenge for many chemists to do in practice. So it's difficult to make CC bonds because they're rather inert. It's difficult, therefore, to have them react and to do that in a controlled fashion. And often you get side reactions, and that makes it dirty, and you have to use all kinds of weird solvents, and so you create all kinds of waste problems. And therefore, there's always a very big step from a lab discovery of how to make a molecule to something that could be eventually practically useful to be made in companies, in big reactors in industry, because then it has to be efficient. Because otherwise it would be a waste to do it. And it requires, of course, highly skilled people to do this type of chemistry. So, especially Sharpless, one of the winners of this year's Nobel Prize, thought, well, couldn't we do that in a more simple way? Couldn't we make big molecules in a way that uses chemistry that can make large molecules, that always works in the way you predict it would work, that works in water as a solvent, so skip all the organic solvents, all the smelly stuff around it, and produces no waste products. And that is what he called click chemistry. So click chemistry is something that basically, if I would say what is the most ideal type of chemistry, well, you pour one reagent in here and one in the other, you just add it together, you wait, and then it's done, you know? And that is the kind of chemistry you would like to have done. In water, nothing fancy, and nothing fancy regarding equipment and stuff like that. So let's look at that. So buckle up for what is click chemistry. So I'm going to give you a little bit of a history to click chemistry and how it came to the development of it, and finally now to the price of it. So it was around 2000 that Sharpless coined this term click chemistry. So he coined the concept of click chemistry, which he said is a form of simple and reliable chemistry, where reactions occur quickly and you avoid unwanted side products. So a very noble way of trying to do that. He was at that time, I think, already in his 50s. And of course, he had already a long life of organic chemistry with all kinds of messy reactions past him. So maybe that was his way of thinking, okay, let's do something radically different now. And he came up, and that was a coincidence with this reaction, the copper catalyzed azide alkyne cycloaddition. Or it's nicely pronounced as quark. So you can use it like that. So that's the abbreviation for this thing. But good ideas often do not come alone. So, and you saw it here, and people have seen it with other big ideas. Two people found that reaction at the same time. So it was published in the same year, just a few months after each other. Actually, Meldal was the first one, a Danish guy who got the Nobel Prize also this year, so for the same thing, and Sharpless himself, so in his lab. They both published this particular type of reaction that was the first, is regarded now as the first and archetypical example of the click reaction. So what is it? It's basically two molecules being clicked together, being reacted with each other, and each have a handle. Each of these two molecules are equipped with an easy handle, so there is a so-called azide group and an alkyne group. An alkyne, for the people who've had a little bit of chemistry, this is a triple bond CC bond depicted here with this threefold lines. And that reacts with these three nitrogens together, this azide molecule, to give this particular connected group, so this buckle, so to say. It's an irreversible reaction, and this reaction can work thermally, but then it takes long and you have to heat it, and you better use also another solvent with that, so that wasn't so convenient. So what both Sharpless and Meldal found is that you can actually catalyze that process with copper. And catalysis is also a well-known concept in chemistry, and it's heavily used in all kinds of industrial processes. A catalyst is a species that promotes a reaction, makes another reaction speed up without itself being converted. And so this reaction is actually stimulated by the use of copper-1. So it's a copper metal ion, so that's used as is called an homogeneous catalyst. So it is dissolved in the same solvent, in water typically, with these reagents, and that promotes that click reaction, so the formation of this bond, in a way that you don't need to heat it, you don't need any additional solvents, it just runs at room temperature within an hour or so. Whereas Meldal did this with really a copper-1 salt and some uncommon side stuff. The big breakthrough of Sharpless was that he could use a very simple copper-2 salt, which are much easier to get, they're much easier to dissolve in water, and he in situ reduced it with ascorbate, which is vitamin C. So it's a very simple common compound, you just add it, and that reduces in situ the copper-2 to copper-1, that becomes the catalyst, and that promotes then the reaction. So it's really like throwing all these things together in one pot. You just stir and wait, and stuff is done. It's as easy as that. So the chemistry to do it is easily done. The mechanism of how it actually works is still a bit complicated, and actually not all the details have still been figured out. So there are still open questions of how the reaction exactly works, and how that, especially the role of the catalyst, and how many units of the molecules are involved in the active site that leads to the final product. But the advantages, and therefore in the end also applications, are plentiful, and labs and companies all over the world are making use of this chemistry. And that's why it has been chosen now as a topic for the Nobel Prize. It's not just that this particular reaction is super fancy or extremely new or whatever. No, it's because it's very useful. It's a very useful tool in order to build upon and use it in all kinds of applications. And one of these things that was actually picked up by the third laureate of this chemistry prize. And she asked herself, So after we can do all this now, so we can make large molecules, it works in water. And if you think about water, then of course you can think of, well, our own cells. We are, as living creatures, are for majority composed of water. Can you use it to do reactions inside living systems, inside living creatures? And that was the next step of click chemistry. And that's where Carolyn Bertozzi got the prize for. She developed click chemistry such that it can actually work inside living creatures. And she developed what is now called bioorthogonal chemistry. And what is that? So orthogonal means that something is separate from it and doesn't interfere with other things. And of course, our cells are very active with chemistry. We are big reactors in a sense, where lots of reactions occur. We have had dinner, most of you here, and so you're converting stuff that was in the food, and that food and energy is being used to create building blocks. We make proteins from that, and etc. There's a lot of processes happening all the time in your body that are basically chemical reactions. So if you want to do chemistry inside the cell, you're basically imagining that you want to do something inside a complicated machinery where already thousands of reactions are happening at the same time. And you don't want to interfere with those. So you have to be sure that you develop a chemistry that actually works inside such a living system without interfering with the whole processes of life. So she started doing that and explore this on individual cells first, and that is basically the biggest breakthrough of and the biggest application area also, I would say, of the click chemistry. So you would think, well, let's take that reaction from Sharpless and co-workers. So could you use this copper catalyzed thing? No way. Copper is actually very toxic and dangerous, especially the copper-1 form of it. So that's not going to work. So what Bertozzi developed was a different type of click chemistry that would run even without a catalyst. And that was, the breakthrough was in seeing that you could achieve that by using alkynes as a reagent that are not this straight or terminal form in which we normally find alkynes, but in which we use a cyclic form like that. So the triple bond, this is I indulge myself in a little bit of chemistry background here. If you have a triple bond chemistry, 2 carbon atoms connected with a triple bond, then the other bonds connected to it will always lie on one line. That is the optimal geometry of such a molecule. If it doesn't do that, you can try to bend it, but that costs energy. So you can actually make molecules as depicted here, where you have a bend around that triple bond to make this cycle, but that costs energy. So these molecules are strained, as they're called. And if you do this reaction now, you release that strain. And that's what promotes this reaction. So instead of using a catalyst, it uses the energy that's basically locked up in that molecule to start with. And then you make a molecule that has no strain in that system, because a double bond can easily live with that conformation, as you see it there. So now this reaction of this alkyne with the azide runs spontaneously. So you don't need to use any catalyst anymore. So no catalyst needed, therefore also no ascorbate needed to reduce copper-2 into copper-1. So it works in water, and it actually works inside living creatures, and it does not interfere with any of the living chemistry. So what did Bertozzi use it for? She actually used a very smart process that's called now metabolic labeling. So if you want to think, and she is a glycochemist, what does that mean? So glycans are sugar molecules that basically are found on the outside of all our cells. And they regulate how cells interact with their environment, how they interact with other cells, how they interact with, try to fend off invaders like bacteria or viruses. And Bertozzi was very much interested in trying to understand what kinds of sugars are there and how are they involved in the dynamic processes that occur in cells. So she wanted to label these glycans in order to see where they go while this cell is living and doing its metabolism. So what she did is she would feed a simple sugar with an azide to that cell. And that cell would think, oh, that's a sugar, I can use that, and I build it in my glycans. And it's actually small enough and doesn't interfere with all the reactions that are occurring inside. No, it's actually simply used. It's seen as a single sugar without the azide. And it's converted just as easy as that. So that sugar is being taken up and then built in the structure. So and that means that you get some of these glycans on the cell actually having azide groups exposed. And then now you can offer a simple alkyne molecule with, for example, a fluorescent dye or a radio tracer or whatever you would like to have as a label there. She worked mostly with fluorescent molecules. And now you can just offer it, and this reaction spontaneously binds to the groups where you have the azides already. And therefore, that reaction then clicks on, and then you can follow by fluorescence where this whole glycan is actually going. Is it staying on the surface? Is it going back inside the cells? Or what is it doing? That's what she did. She made pictures of cells, and in green you see the fluorescent dye that is clicked on two glycans that were labeled this way. And what you see is that these glycans are not only on the outside of the cell, so you see many on the surface of these cells, but you see also little patches of glycans that are being taken up, for example, by some uptake process. If there is a particle or a virus particle, then by endocytosis, things are being taken up and brought inside the cell. And this usually goes together with some lipids, with some glycans, and that process can then be visualized by these fluorescence things. So you can study this way the dynamics of how cells behave. And that was, I think, one of the breakthroughs for which Bertozzi got the prize. So what else can you do with click chemistry? So it's now also being used, for example, to make drugs. So this is a drug that is based as an immune therapy drug. So it uses an antibody and then connects a drug to that antibody via, you see here, that product of click chemistry. So basically you have a side group here that you can attach to the antibody with an alkyne, and then you have this drug molecule you design with an azide, and then you can basically click the two together and you submit it. You inject it into a patient, which then brings this drug close to where the antibody binds. So if the antibody recognizes specific types of cells, that means that you can actually target the drug to specific places where you want the drug to be. Same is true for cell therapy. So there are ways of similar ways you can label cells, as we've seen also in the work of Bertozzi. So you can label cells this way and attach drugs to it, attach growth factors to it that would regenerate tissue and stuff like that. So a little bit on to the people behind this work. So I already, of course, showed a bit in the history of these people, but so the prize was equally shared between all three. So Bertozzi, Meldal, and Sharpless. And so Bertozzi, and you see here a picture, she's the youngest of the three, so born in '66, US born, studied and got her PhD in Berkeley, and is currently at Stanford University as well as at a medical institute. She's very active, as I said, particularly in glycobiology, so trying to understand how sugar-based molecules that are active in cell processes, how they work and how that affects living creatures. She got various prizes, amongst which also prizes from our Dutch community, so including the Heineken Prize and Bijvoet Medal, etc. Meldal is, for me, it's a bit of I didn't know him, so the other two I've met at meetings, so Bertozzi as well as Sharpless, and they're extremely famous. Morten Meldal is a person who is most active in peptide synthesis, which is a bit out of my own area of expertise. Still very productive, but he's very focused on how to make peptides. And his original paper with this click chemistry was how to make peptides in a more efficient way and easy chemistry using that particular click chemistry. Sharpless, I think, is the most unique or the most creative of the three. He is truly exceptional, and that was also recognized. It's for him the second Nobel Prize in chemistry. He got it already in 2001 for a series of name reactions, so reactions named after him that he developed in the time before: Sharpless epoxidation, Sharpless asymmetric reactions. So very much he worked on synthetic procedures in the past to make chiral molecules, molecules of one particular type of handedness. And he was awarded with the Nobel Prize, as I said, in 2001 already. Very early on in his career, actually, he had an accident in the lab which cost him the eyesight of one of his eyes. And that's why he has this quote, and that you should always wear safety glasses in the lab. There's simply no excuse not to. So, and that's he learned that the hard way, but didn't prevent him to have to think of very brilliant ways of how to do chemistry. So he is really amazingly creative in thinking how you could design new reactions and how to do that. And so he has developed numerous new types of reactions that we didn't have before. So he's in an exceptional route. There were only 4 people before him who had 2 times the Nobel Prize. And there are a couple of organizations who got it more than once. But you see here, especially, of course, noticed is Marie Curie and Linus Pauling. They have had the prize twice. But in chemistry, 2 times the prize of chemistry was actually Sanger and now Sharpless. So, yeah, click chemistry. What else? So that's the conclusion of my talk. Thank you very much.
Thank you very much, Jurriaan. That was an exceptionally nice and clear talk. And we have time for a few questions. Yes, please.
Please, do you think that...
Yeah, hold on. You can get a microphone. Yeah.
Do you think that bacteria use click chemistry?
I don't think so, to be honest. So I think none of the living systems are actually using click chemistry, because otherwise it would interfere with living chemistry. And so these compounds are typically not found in living systems at all, and that's why you can actually combine them with living systems and do chemistry there that doesn't interfere with all the other many functional groups that are around in living systems. So yes, you can apply the technology to bacteria and to have them do things, and there's a whole area where people now, like triggered by what Bertozzi did with feeding cells with artificial sugars. There's now a whole area where people try to feed cells, bacteria, whatever, with artificial feedstock, which they would normally not use in natural ways, but which you can use in order to make produce all kinds of molecules that are of interest for us. So we can, for example, try to train bacteria in order to have them make drugs for us that they would normally not do. But you can, if you feed them in the right way, and if you change their genetics also and optimizing that particular pathway, you can actually get productive factories out of living systems that way. Yeah.
Yes, please.
I was wondering if you could make like any molecule with this method, because I can imagine that the extension that you have to add to the 2 molecules in order to click changes the behavior of the resulting molecule in some way.
No, you're fully right. I mean, it's not, you cannot use it for every molecule. If you really want a very particular structure of that molecule, you still have to make that molecule. But very often, chemists need something simple. They want some functional group they can make separately, and they want it to hook it up to something else. And then that link can be easily made with click chemistry. And that's what I think I mentioned it on the slides. I didn't say it explicitly, but it's called functionalism. So many chemists think of introducing a function inside a system. And then the structure of how you achieve that function is not so important. So you can find different ways of achieving that function. And then you might find ways where click chemistry can actually be useful to do that. So no, so you're right, you cannot make every structure with it. If you want to make Taxol, you cannot do that with this click chemistry. But you can make a whole lot of functional molecules using click chemistry. That's absolutely sure.
Yes, we have 2 more questions, and then...
Thank you.
Yep.
Maybe a natural question could be, is it possible to unclick them? Because when I look at the pictures, for example, in the presentation, it looks like it is possible, but I really wonder if it is the case.
I don't think so, to be honest. Yeah. So we actually use it a lot in our labs, but they're very stable. So they're very stable products, and they're designed that way. And it also makes that they run that quickly, because it goes downhill, so to say, in energy quite a lot, and that the products are stable in the end. And in most cases, again, that's useful. There's a whole... nevertheless, these things do spark people with new ideas of also trying something like this. So designing chemistry that can be reversible if you want it. But then you need, again, other types of chemistries. And these are there now. They're, I think, more recent developments, maybe another Nobel Prize in some years from now.
We'll ask you again then to introduce the unclick chemistry Nobel Prize, and we go here.
Well, obviously it's really handy to have these click groups, but I still see a problem of that you need to attach them to the proteins you actually, or not the molecules you want to connect. So can you maybe go into detail how you solve that? Because it's still difficult to just like merge things together, right?
Yeah, so it's a good point. So it depends on what you want to achieve. So let's focus on proteins, for example, because that's where things do happen a lot, or sugars like these. There are various ways of introducing functional groups on proteins. So there are various types of reactions which could provide these anchors. Some ways are, I would say, brute force. So for example, most proteins have functional amine groups on their outside, and usually many. And then you would just add a reagent that reacts with a couple of those, and you don't even know exactly which ones will actually react, but some, maybe 3, maybe 5, maybe 7, you don't know. And then you would have a couple of anchors to which you can put a dye. If you can live with that product, if that is your functional system, fine. If you want something more specific, then you might need to rely on different methods. For example, there are methods to, by genetic methods, put in different amino acids into the protein. And so basically you bioengineer your protein, so to say, with a different amino acids. And that amino acid could be an amino acid like the azide sugar shown in the feeding of glycans. It could be an amino acid with an azide. And that is actually currently being done. And then you introduce at a very specific location of the protein, you introduce your handle. And that protein can still be fully functional, because usually there's one side of the protein that does its job, like recognizing something or converting something, like enzymes do. But the rest is surface that is there for interacting and stuff. And that's usually a good place to put a handle and then put a functional group there. So there are various ways of solving that.
Thank you.
Yep.
All right. I have a last very brief question. So you mentioned that vitamin C is used to reduce this copper. So what is so special about vitamin C that that's the molecule of choice? And connected with this question, is that also, let's say, the main function of vitamin C in the body to reduce, let's say, systems? Or
well, vitamin C in the body does a lot.
A lot more.
Yes. Okay. and it's used here as, I would say, as a nice and green and convincingly easy to use chemical, so to say, to promote the chemistry.
It could have been some
it
other
could have been
acid
something
or,
else,
okay.
without doubt.
Good.
Absolutely, yes.
Okay, well, thank you very much, Jurriaan Huskens, and give him a big hand. For the Nobel Prize in Physiology and Medicine. And we now continue with the Nobel Prize in Physics. I'm not sure if this microphone is now on. Yeah, should be. Okay. Please, Pepijn, come up on the stage. So the Nobel Prize in Physics is not about quark, but it's about quantum. So it's somewhat connected maybe. And it will be introduced by Professor Pepijn Pinkse, who's a professor here at the University of Twente in adaptive quantum optics. He studied in Leiden and did his PhD at the University of Amsterdam. So we have a lot of connections now to other universities in the Netherlands this evening. Then he went to Germany for a postdoc at the University of Constance. After which he became a senior researcher at the Max Planck Institute, another Max Planck Institute for quantum optics, this time in Munich, where he worked for more than 10 years. So he really was very well embedded in this institute. And since 2009, he's back in Twente. He was the program director for the Applied Nanophotonics program within Mesa Plus, which helped to develop the very strong nanophotonics cluster that we have now here in Twente, one of the biggest, let's say, conglomerates of photonics people in the Netherlands. And since 2019, he is positioned here as a full professor. He is also one of the laureates of the Fiji Award of NWO, I think in 2012, if I'm not mistaken, and was also co-founder of the Quix company, which you may have seen in the news. It's a company about quantum technologies based on optics, and that's also the topic of the Nobel Prize in Physics for this evening, but Pepijn will tell you all about it. Please, Pepijn.
Thank you very much, Hans, for the introduction. And thanks for the invitation to introduce the Nobel Prize of Physics here this evening, which is an honor. I met two of the gentlemen, Alain Aspect and Anton Zeilinger, on several occasions. And it was really a pleasure to be inspired by them. So if you read the announcement of the Nobel Committee, the Nobel Prize in Physics was given for experiments with entangled photons establishing the violation of Bell inequalities and a pioneer in quantum information science. This is a mouthful of complex words, which I think many of you have never heard of before. And my job for the next 20 minutes or so is to explain some of these words, in particular the word entanglement, in the word entangled photons, Bell's inequalities, and what we do with entanglement photons in the quantum information science. Okay, let me start very basic with what is the quantum mechanics. How was it in the early 1920s when the whole story starts? Planck and Einstein introduced the concept of quanta. We know electrons, atoms, molecules. And they introduced the concept that you describe these particles, maybe we should put this microphone away, by wave functions. And one of the quanta we're going to talk about today are photons, which are already waves, as we know. But important for this story is that you should realize that light, as we know it, is an electromagnetic wave. So indicated here, there's an electric field that is oscillating in a particular transverse direction. So if light is moving towards you, then the electric field is moving up and down, or left or right, doesn't matter. But it's in a transverse plane that it's oscillating, and there's also a magnetic field which is oscillating in the orthogonal direction, which is not important for this evening. And if you take a small amount of energy with a discrete amount of energy, we call this a photon. So it's a quantized packet of energy of light. These are the photons we are talking about. And now, some of you might have heard about the Stern-Gerlach experiment, where you send atoms with a spin through some magnetic fields, and they always give one out of two answers. Something similar happens with photons. If you send a light beam, or actually single photons, doesn't matter, onto a beam splitter, a polarizing beam splitter, we call this. This is a glass cube with an interface in between. It has the property that light that is oscillating, let's say, vertically is transmitted, and light that is oscillating horizontally is reflected. Now, if you put a single photon, if you send this towards this beam splitter, it cannot be split. There are two single photon detectors behind it, and only one of them will click. So you get detection event. Of course, if the photon was polarized vertically, then this detector will click. If it was polarized horizontally, this detector would click. Maybe Jurriaan, you might like this. We have click physics here. Yes. The most natural thing to think about would be independent photons. So let me start with a source here, which emits random independent photons, meaning that if the photon going to the left could be horizontal or vertical, the photon to the right can also be horizontal or vertical. And if you look at the statistics of these 4 detectors that click, then we find that, well, if this is 50% horizontal, 50% vertical, and the one to the left as well, then the chance of finding 2 horizontal photons, so that this detector clicks and this detector clicks, is 50% times 50% is 25%. And so you can fill out the whole table. Every of these combinations is equally probable. And now we come to the entanglement. So there are sources, and I give you an example later on, that emit photons that are entangled. We say that these photons are entangled if they, for instance, are in a state where they're both polarized in the same direction. We do not specify which direction, but they will be polarized in the same direction. If you then do the statistics with these 4 detectors here, we find something remarkable, namely that if the one on the left indicates that photon was horizontal, then automatically the photon on the right also was horizontal. You will never find a vertical horizontal or a horizontal vertical case, but you do find vertical vertical. So, and actually, if you would place this whole unit of polarizing beam splitter and detectors under a certain angle, and this one at the same angle, you would find exactly the same diagram. So the only property of this source is that the photons are emitted in the same polarization, in an entangled state, where we do not yet know what that polarization is, and we only know it at the moment that it's detected. But now this leads to some difficulties in understanding, because what happens if you send one of these photons to Jupiter, one to Venus? That means at the moment that the photon is detected at Venus, you automatically know that the one at Jupiter has the same property, for instance, horizontally polarized. Well, that's weird, because how does the other photon know that its other partner was detected in a certain direction? Well, this is conceptually problematic, and if you find this problematic, you're a good company, because Einstein did not like this at all. So this is a famous paper from 1935 by Einstein and 2 junior colleagues, Podolsky and Rosen, where they wrote that this collapse of a wave function, as it's described in quantum mechanics, or affected by measurement at distance, that cannot be real. I mean, this is too outrageous to think that it's true, although they acknowledge that it predicts the experiment in the correct way. And Einstein, Podolsky, and Rosen said, well, there must be some other theory. There must be some additional properties to these photons, once they are entangled, that will predict the outcome of a measurement later. And they call that local hidden variables, meaning that there's something local to a photon, so that property travels with the photons, let's say to Jupiter or to Venus, that will predict the outcome of the measurement. They don't know how this information was stored, but Einstein, Podolsky, and Rosen were convinced that this should be the case. So think of some hidden rule book that the photons have discussed before separating, saying, for instance, that if you are measured with the polarization detector under 45 degrees, then you should act as if you were horizontal. Or if you are measured under 90 degrees, then you should act random. Or whatever rule you can think of, locally defined. Einstein and Podolsky and Rosen were convinced that there should be some set of rules like this that would predict the outcome or preset the outcome of these clicks at the detector. Well, you might think that is all nice, philosophical. Who cares? Well, John Bell cared. He cared a lot because he realized 30 years later that this is not only theoretical and academic discussion. He proved that so in the famous paper which was on the Einstein-Podolsky-Rosen paradox, that whatever set of rules you come up with, so whatever local hidden variable theory you can think of, it will make a different prediction for a certain set of experiments than quantum mechanics. Meaning that if there is now a possibility to do an experiment which shows that quantum mechanics is true, or there is some local hidden variable theory. So to understand what Bell's theorem was about, you have to again look at these photons coming from a source which emits entangled photons. Actually, we call this a Bell state. So photons that are, for instance, polarized in an identical way, although we do not know yet how. That depends on the measurement. And in deriving Bell's theorem, we are interested in properties the probabilities that, for instance, this detector fires and this detector, or this detector and this detector. You can make all kinds of combinations between these probabilities of pairs of detectors clicking, and then you can do statistics on this. Well, one additional requirement is that we assume that the source has, that the detectors, there's no prior bias, that if you only look at, let's say, the detectors on the left, they detect basically random. 50% of the chances, 50% of the cases, one of them clicks, 50% the other one clicks. Same thing on the right. There's no prior bias. If you now choose these angles of the detection systems to be the same, then the prediction of quantum mechanics says that if you measure a horizontal photon there, the other one will click as well, and never a vertical one there. And if this one clicks vertically, then also this one clicks. And we know this should be the case because actually what angle the detection systems were set should not matter because the system is set up to have no special angle involved. Okay, now in contrast, we have the prediction, as Einstein, Podolsky, and Rosen predicted, which has some local hidden variable rule book for these photons. You can make all kinds of choices for what kind of rules you let the photons behave like. Give one example here. Let's say that we set the detectors under a 45-degree angle, and then we look at the statistics of these detectors clicking, and we see that because the photons here, they are preset, they have some local hidden property, they're either horizontal or vertical, and if that's because that's 50% of the cases, the probability of having HH is 50 times 50% is 25%, and horizontal or vertical is 25% of the case, etc., etc. They are all equally likely. Now, we just would derive the property, the same statistics for the quantum mechanical prediction. Oh yeah, a different table. Well, you can say, well, wait. You chose a very particular case. I could have sent out photons with classical properties that are either diagonally polarized or anti-diagonally polarized in the same way. Then yes, those hidden variable photons would give exactly the same table as the quantum mechanical prediction. But then there would be other sets of detection events which were not coinciding with the quantum mechanical prediction. And this was the brilliance of Bell, that he came up with a certain combination of probabilities that would always would always be different from a local hidden variable theory and a quantum mechanical one. And then Clauser, Horn, and this is the Clauser from the Nobel Prize, Horn, Simony, and Holt came with a slightly more practical and more robust version of this inequality. And the inequality defines some parameter S, which is some combination of probabilities that you can actually measure in the laboratory. And they, based on Bell's inequality, the new version is now also called the Bell inequality in the CHSH version, that the absolute value of this parameter is always smaller than or equal to 2 for all possible local hidden variable theory. Well, and Clauser, he was there. He realized that he could actually do an experiment. So what Clauser started doing is come with a source for entangled photons. Where do you get entangled photons from? And Clauser came with the idea, well, let's use calcium. Calcium is an atom that if you excite it with some UV light, it will quickly decay to some other state, and actually the details of the states are not important. From this state it will decay with a green and a blue photon, and these photons have this property that they are always in an orthogonal polarization, and then you can convert this into, for instance, the state that I discussed before, where the photons have the same polarization. Then he did an experiment. There was an oven here. These atoms were sent on a beam. In the interaction zone, they were excited by some arc lamp, and then photons were emitted in pairs and detected on the left and on the right after some polarizers. And then if these detectors on the left and on the right clicked, that was then registered by some coincidence electronics. And obviously this proved beautifully the predictions of quantum mechanics and ruled out the predictions of the LHV, the local hidden variable theory. However, because this distance of the detectors was 5 meters, and the polarizers were basically fixed, this meant that critics said, but at the moment that you emit the calcium ion emitted these photons, they already knew how they were going to be measured, so they could have made some agreement on how to be detected. So this is not very conclusive. What you actually should do is you try to decide how to measure them after the photons were emitted. And this is where Alain Aspect came about. He and his colleagues Philippe Granger and Roger did an experiment in 1981. Actually, you can easily recognize Alain Aspect. He always has this nice, beautiful French moustache. A very friendly guy. Talked several times to him. He made an experiment where the direction of the detection system could be switched by some electro-optical switches. So there's one here, C1 and C2. And the switching electronics mechanism was so fast that you could actually switch or decide on how to measure what angle these photons were measured after they were emitted by the source. So whatever they made up when they were still in the atom, they could not have used the information about the detectors because it wasn't decided at that moment. And of course, this also proved the prediction of quantum mechanics with few statistical error bars. Well, then the third Nobel laureate here, Anton Zeilinger, very big Austrian erudite man. Many contributions to all kinds of quantum optics. He and his colleagues, Paul Kwiat and Harald Weinfurt, I also met. They did an experiment with improved statistics, with much higher rep rates, much higher detection efficiencies, that showed that the outcome of this Bell inequality. Remember, Bell said if you have a local hidden variable theory, S should be smaller than 2. They measured 2.6489 with only uncertainty in the third digit. So they absolutely ruled out that by statistics they were wrong. Okay, are we happy now? No. Yes, this proved that Einstein and Podolsky and Rosen didn't believe in this LHV, in the quantum mechanical prediction, and that they thought that LHV, local hidden variables, should also be able to predict, but because of these experiments, we now know that no, reality is really described by quantum mechanics and not by this classical LHV theory. However, critics might say, well, you have loopholes. The efficiency of your detector in your experiment was not 100%. So maybe those events that you missed somehow contrived to make you believe something which would not have been there if your detectors were perfect. Okay, well, hard to believe. And how could the photons know in advance how to fool me? But we can solve this. How? Okay, I guess you can think of some solutions. You just make better detectors. Getting closer to 100% is easy. It's more difficult to fool the experimentalist. You could also use particles that are easier to detect. For instance, atoms are easier to detect than photons, for instance. Okay, so this loophole has been closed. People have done experiments with very good detectors. But then you say, well, these detectors were here and there, and they were very efficient, but they were pretty slow. In the moment that this detector clicked, a signal could have traveled to the other detector and influenced how this detector was measuring. Okay, and also there's a simple solution. You place the detectors very far away and you make them very fast. So that even the speed of light is not enough to make the detection event on one side influence the detection event on the other side. And to every sensible human being, this should be enough, I would think. And each of these loopholes has been closed one by one until 2015, not simultaneously, but why would you, I say? I mean, if you find spooky action at a distance hard to believe, then I find it even harder to believe that nature somehow conspires against humanity to pick the detector loophole in some experiments and the speed of light loophole in the other experiments. I find this personally very hard to believe, but anyway, people did close the loopholes simultaneously. And one of these, the first experiment actually was done here in the Netherlands, in Delft, by the group of Roland Hansen. A beautiful experiment where they had a light source and that they had systems based on spin qubits in diamonds. Details don't matter, but they can be read out with very high efficiency. And by placing these detectors very far away, he could actually have both high detection efficiency and be so fast that the speed of light could not have fooled this. So this is the first Dutch connection, obviously. There were two other experiments in the same year. One in the University of Vienna by Anton Zeilinger's group, and one in NIST Boulder, Colorado, that basically, with other technology, but closed both loopholes at the same time. So it was really a race where there was only a few months difference. If you look at the author list, there's something peculiar. I'm not sure if you guys from the group know, but there's one person that was an author on all three of these papers. Not sure if you can spot it. It's this person here, Morgan Mitchell. He was in all three, so you could say there was a Morgan Mitchell loophole, and maybe he did something wrong in all three experiments. No, but so his contribution was that he actually he was the expert on the random number generator that was used to randomly set the angles of the detectors. So this was a crucial contribution, and he is in all three of them. Okay, where is it good for? Well, entanglement is actually a resource. We use it every day in the laboratory. You can use it for quantum teleportation, which is in by itself now a resource in quantum computing, and you cannot do quantum computing without quantum entanglement. And actually, quantum teleportation is the last topic I would like to mention because it was also pioneered by one of the Nobel laureates, maybe Anton Zeilinger's group. Quantum teleportation is the process where you have some unknown quantum state. Let's say Alice has this quantum state in her possession, and she wants to send the information of the quantum state to Bob without actually sending the atom to Bob. That would be trivial. And for that, you cannot clone quantum information, so you need to do something locally with Alice's unknown quantum state, and then send something to Bob. And the way this works was suggested by Bennett and others in 1993. It involves a Bell state, one of these entangled states. One arm of these Bell states is then measured together with the unknown quantum state from Alice, and actually it's lost in that process. Then some classical information is sent to some device at Bob's side, where the other half of the Bell state is actually rotated in a certain way, and then you can prove that if this measurement was successful, that the state is teleported. Well, you immediately recognize that this has nothing to do with this kind of teleportation. There's no matter moved, there's only information. And worse, this process only works in 25% of the cases, but in 100% of the cases, the original state is destroyed. I would not like to step in front of one of these teleporters where I know that I have 25% chance of getting out on the other side. So this is actually used a lot in quantum information theory. So this was the paper by Anton Zeilinger, and this is now the second Dutch connection here. The first author, Dick Baumeister, he's a professor now in Leiden University in Santa Barbara, USA. Okay. Where are we today? There are quantum computers based on small ones, ions, atoms, photons, superconducting qubits, circuits, quantum dots. Most of these quantum computers are so small that they can still be simulated by classical computers. However, there are experiments, several now at the world, something like 2 or 3, that have reported the achievement of a quantum advantage. A quantum advantage is a difficult word for saying that these devices have done some computational task. We don't know yet for what it's good for, but there is some computational task that is provable hard for classical computers to redo. So at least at this moment, I mean, you can no longer deny that there is some computational edge to quantum computing that cannot be reproduced by classical computers. I think this is a really big breakthrough over the last few years. Entanglement is used routinely for certain types of quantum communication and also quantum sensing. And back to the quantum computers, there are actually several quantum computers in the cloud. Can you guess how many? I mean, if you would ask me a month ago, I would have said maybe 2 or 3. There are already 13. And they're big names here. So IBM Q was the first one. Actually, we use them in our lectures of quantum information science, quantum information processing. Students will learn to program a quantum computer, for instance, the ones from IBM. There's an optical one in Canada, Xanadu. There's a Chinese one from the Alibaba group. Google stepped in, Amazon stepped in, Microsoft stepped in. There's a Dutch one since a few months in Delft. And we have good hopes that actually by next year we will also have one here in Twente. Okay, last slide on what we do ourselves. So I work here at the university. We use entanglement and quantum information science daily to do basic research. So some results that, for instance, Frank worked on is to experimentally demonstrate some experimentally unproven results from quantum thermodynamics. We worked on things that we call witnesses, and this is all done by an optical integrated photonic processor that is built here in Twente. And this is all done because of the groundbreaking work of, I should call them actually all 4, because John Bell unfortunately died too early. He was in the early 60s. I think the field is convinced that he should otherwise have deserved the Nobel Prize many years ago. But certainly I should attribute this to Clauser, Aspect, and Zeilinger. I'd like to thank you for your attention.
Thank you very much, Pepijn. Looking at our shirts, I feel that we're also kind of entangled.
Entangled, yes.
But I guess you also got a phone call from Mitchell Morgan this evening, what to wear.
Not yet, no.
Okay, okay. Well, then maybe there's still quantum things playing a role. Okay, we have time for a few questions. Oh, all the way in the back. That's very good. I almost want to throw the mic, but that's maybe not a good idea. If you can pass it.
First of all, thank you for this amazing lecture. But I have a question about quantum computing. As most of the companies right now in the industry especially are using superconducting qubits as circuits. And these experiments have shown that you can have a system of photons in an entangled state and controlled. I would like to know if you think that this might switch the focus of the industry or the research into more of photonic circuits than the superconducting qubits.
Oh, thank you very much for the question, because indeed the first quantum computers online, the IBM Q for instance, were based on superconducting qubits. However, the Xanadu system is optical based. The one which we want to put in the cloud will also be, of course, optical based. There's one other big company, PsiQuantum, which is not on the list because they have nothing in the cloud, but we all know that they have investments of several $100 million to develop a optics-based quantum computer. So I think there's really a race going on now, and it's too early to say who will win. Maybe there's room for both platforms, both the superconducting and the photonics, to win. Yeah, it's very exciting times. I can't answer more than that at the moment. Yeah.
Other questions? Yes, please. And maybe I can ask one question first before I get there. So if you take, if you do this Bell test, you basically you measure statistics. Huh? Do you have to do rotations of these polarizers and then add up all the statistics? So you cannot really fix the polarizer.
No, you have to do measurements in at least multiple two bases.
Okay.
Otherwise, there's always LHV theory which can do the same result.
Here, yeah.
Please, could you tell something about photo-quantum thermodynamics?
Oh, I do not have a slide on this, but so the experiment that later on you can talk to the student who did the experiment. So, the idea there is that there's thermodynamics, which says that if you have a cube of ice, it will be thermalizing with the environment, and it will melt. It will be a puddle. We all know that the arrow of time will go in one direction. It will melt, and it will never spontaneously form a cube again. On the other hand, quantum mechanics is a reversible theory. It says that you can always invert every process in time, which seems to contradict thermodynamics. And theory predicts that if you have a finite system, then you can say, okay, this is my bath, and only this is my system of interest. And you can see that the system of interest will thermalize, and it does so while keeping the entire system pure, because entanglement is building up. And with the photonic processor we have here, we could actually show that the small system thermalizes, and then by undoing the transformation by putting more optics behind it, because we have so much control, we could see that we get the original pure state back. So that is, and actually you can Google the archive paper and you can check for yourself the details.
Frank was his name? Frank?
Yes.
Frank, you should be describing this in one of the future Nobel Lectures, or maybe also in a colloquium in the Studium Generale. So please be prepared. The last question, and then we go to the third Nobel Prize. Yes?
Okay, thank you. I have two questions, if I may.
If it's short?
Okay, the first question is, it's very clear that there is a lot of effort to show that this local hidden variables theory is false. Mm-hmm. But at the same time, we have quantum mechanics, which is clear that performs better than that, right? So what's the problem of quantum mechanics that makes people try to show that the local hidden variables is wrong? And the second question, in the articles that you showed, we saw that the previous experimental work was possible to do with small groups, 3-4 people maximum. But if you look at the new ones, it's impossible. There are too big teams, right? Was it the reason that at that time there is a problem of recognition, so that there are some hidden scientists, in fact, which are not named in the articles, or are we in an era where we cannot think a quantum experimental work with small groups?
Okay, let me answer your first question first. I agree. I mean, I completely believe that the predictions of quantum mechanics are true, and I don't have a problem with that. But I also understand why Einstein, not the least of our physicists, had a problem with this, because if you try to explain with your classical mind the predictions of quantum mechanics, then you get into trouble. But you don't need to. I mean, you can use the quantum mechanical language. So it's harder to say anything more about that. On the size of the groups, I mean, I do not agree that you still need, that you cannot do experiments in quantum computing, for instance, with necessarily why it can't be a small group. I mean, I think Frank did the experiment basically on his own, or maybe one other student. So yes, there are some experiments. For instance, IBM, the Googles, they have big teams because there's a lot of nano engineering that is needed. But I don't think this is a requirement. You can still do small physics with beautiful physics, nice results with small teams.
So after dark matter and dark energy, there's no need for dark scientists, right?
No.
Then let's thank Pepijn again for this nice talk on a complex topic, and it's a great pleasure, and we really thank Jurriaan for the efforts to also introduce the Nobel Prize for Physiology and Medicine, so that these three prizes that we feel are connected with the science and technology area are introduced this evening. Let's have our attention for Jurriaan for the third Nobel Prize. Please, go ahead.
Thank you. So, what was the prize given about? It was actually for figuring out what is the genomic DNA of our human ancestors. So, for figuring out how the DNA looks like of the Neanderthals and of other earlier human forms. So, and the technology that was used, that's what I'm going to introduce first, because that took the guy who got the Nobel Prize for this a couple of decades, actually, to work out. So, and it's a technique that he did not invent, but he applied to this particular problem, to figure out what is the DNA of our ancestors. And that technique is sequencing. So, and it's actually called now whole genome sequencing. It's the technique that has boomed and boosted over the last few years. And it's a process trying to determine the DNA, all the DNA of a full species, of a full organism, in one go, in one experiment, basically. And so that's, and typically all living systems have two types of DNA primarily in our cells. So we have the DNA that is locked up in our nuclei, and that is basically the carrier that we get from our parents, and that carries all the information that we need in order to build our own cells and to continuously have all the metabolic processes in our cells working. But there's also DNA contained in the mitochondria, and these are the little factories found also in our cells. And the workhorse of this technique is so-called PCR, polymerase chain reaction, and that's chemistry again. So, and actually it's a kind of chemistry that got the Nobel Prize already in 1993. That got the Nobel Prize for chemistry to Kary Mullis. And what does that do? It's a technique by which you amplify very small amounts of DNA. And that seemed in the end particularly useful for the Nobel Prize of what I'm introducing now. So how does that work? So to sequence DNA, you have to think that you need to extract DNA. DNA is typically in this, what we all know, I think, from textbooks, this duplex type of structure. So it's this helical form. Basically, there are two molecules, long strands, long chain molecules, interacting over a very long section with each other, and they contain base pairs. There are only 4 bases, A, C, G, and T, that encode for everything that we need in our cells, and that defines who we are, what we do, and etc. So if you can read out that information from the DNA, you understand what happens, and you can learn what processes can occur. So, but these molecules are big, and they're in one cell. There's basically one set of nuclear DNA that contains all the genetic information that you have. And most chemical methods and analytical techniques to detect are not sensitive enough to do that. And that's why you need amplification strategies, and that's what PCR is for. So what you see here, and that is just in a few words, make thousands of copies. Well, that is what PCR does. So that's a chemical process by which you have these two DNA chains thermally separated, and then you feed an enzyme that basically makes the complementary copy to both of these chains. You feed it with 3 nucleotides, so the isolated A, C, G, and T bases, and they make of both the complementary copy. Then you thermally separate those again, and you do the whole process again. So every time you duplicate the number of DNA chains that you have. And that's what PCR stands for, polymerase chain reaction. So it zips basically those chains together. You make every time, you make twice the number of DNA chains you had before. And that way, if you do that a couple of cycles, 20, 30, 40 cycles, you have, in one go, you have then a whole bunch of DNA, which is now much easier to analyze of what you have. So that's how PCR works. And then you have to analyze basically what you have, and there are various ways of actually sequencing what you have. Now for genome sequencing, it's a little bit more complicated, but because genomic DNA is enormously long. So genes are millions of bases, so they're extremely long molecules, and typically what you have to do in order to make that chemically processable is you have to chop them up. So basically you chop them up, you multiply all these copies, and then you get isolated fragments of which you know the sequences, and then you need the data analysis to overlay certain sections as is depicted here. So you might pick up this fragment or that fragment and that fragment, and then you see that there are certain sections that overlap because they belong to one fragment and the other one was fragmented on a different place. And that's a way, by data analysis, you can basically then depict what the whole chain looked like before you fragmented the thing. So this, in order to do that to whole genomic DNA, that is now, it's a puzzle that has been solved in the past few years. So, and yeah, actually you can have your own genome analyzed by a company if you want to. It only costs about $500, and then you can have your whole genome sequenced by a company in about a day. So it's not so bad. So, and that was a whole development over the past 15, 20 years or so. So now, this we can do with living DNA, with us, with ourselves, with living systems. But now, could we analyze the DNA of Neanderthals? Of course, there have been finds of older people and other species around. And then we come in other technical issues. And that is, of course, where one of the challenges is can we actually find ancient DNA, and is that DNA still complete enough? Because DNA degrades. It is consumed by enzymes, by heat, by UV light, and whatever. So there's a variety of processes that will degrade the DNA. So if you find, for example, bones of older human species, can you still recover enough DNA in order to do that kind of analysis? And can you trust that the DNA that you find is actually from that species and not from some contaminated source, because maybe some animals have been gnawing on those bones before, and so there's a whole lot of stuff to think about. And that was a process that was set by Pebo, that's the guy who got the Nobel Prize this year for this work. And he started basically in the late 1980s already with his work. He really had his mind set on, I do want to analyze this DNA of humanoids. Can I do that? So he was already involved quite early on when PCR started in the 1990s. And he had his first breakthrough by analyzing the mitochondrial DNA of Neanderthals. And why mitochondrial DNA? Because it's much smaller, and there are many, many more copies of mitochondria within one cell than just that single copy in the nucleus that is there. So it's much easier to find good mitochondrial DNA, and that's he started analyzing. And then he started analyzing the differences in genetic information, so these bases that were there, and comparing human to human, so various human samples, and they would vary to some extent from one to another, and also human to Neanderthals, and you see there's a larger variation, and also humans to chimpanzees. And then you say, well, yeah, I could have figured that one out beforehand, because I know that Neanderthals look a bit closer than us, and chimpanzees look a bit further apart. And yeah, this work shows that that's true, but hey, what's the fun of that? So good technological process, and then came the first big hit a lot later, about 20 years later. So he has been, in all this time, kept working in this area of trying to figure out all the techniques in order to do this reliably, get the samples from individual bones and do this techniques. And then he finally was able also to analyze the genomic DNA of Neanderthals. So this ended in a big paper in 2010, where he found that Neanderthal DNA actually overlaps with human DNA. So for about 1 to 4%. Before he started doing this, the general belief in the field was, well, you know, we have some common ancestors between Homo sapiens and Neanderthals, but they branched off some while ago, and there was a period that both coexisted, but the Neanderthals did not survive, and we did, and that's, you know, how the whole situation occurred. But what people did not realize that these species were not only overlapping, but interacting and interbreeding. So there was genetic information flowing, as they say it, between Neanderthals and Homo sapiens, because they were at the places where they coexisted, they were able also to have offspring together. And that's seen in the, again, analysis of how the DNA of Neanderthals overlaps, and especially overlapped with non-African Homo sapiens, so in the areas where Neanderthals locally overlapped, coexisted with Homo sapiens. And that happened across millennia. So there was quite long periods where both species coexisted and also had joint offspring. A next big hit was in the same year. Actually, they started sequencing, and it was immediately a success, a bone of a new find in a cave, in the Denisova cave, somewhere in Siberia. And from the DNA analysis, they could not find any other known comparable species. And it basically meant that the DNA and the bone they found was from at that time, unknown hominoid species. So they called it the Homo denisova. So that was again a different species, and also that turned out to be overlapping in time and locally and interbreeding with Homo sapiens, but now especially somewhere in the areas of Melanesia and Southeast Asia. And up to 6% of those people's DNA currently contains DNA that overlaps with the Denisova humanoid. And those two were really the big hits because they changed how people had to look at the field of how human development occurred and how new species of human forms coexisted and moved across the planet at these very ancient times. So it gave much better insight in the coexistence of species, how they moved, especially of different human species, of course, across the planet, how they interbred and stuff. And it established a new field that's now called paleogenomics. So the genomic analysis of ancient species and how they behave. So the only winner of that prize, so it's one full prize awarded to Svante Pääbo. There's a Swedish guy who, as I said, devoted his whole life in solving this particular question, and he got quite far. So I think it was really fascinating to see it, and I hope you enjoyed this little exercise, which is a bit out of my own expertise, but that's it. Thank you.
Thank you very much. That was very nice and introductory, and a lot of chemistry still involved. And PCR is, of course, very important also in the corona test.
That was the absolutely. It's a standard technique now in many labs.
All right, we have 5 minutes before 8, and 8 we should close. So, who wants to ask Jurriaan? And keeping in mind that he took the effort, but it's a bit out of his comfort zone, maybe. too.
I was wondering, how can you see the difference between a common ancestor in the DNA or interbreeding between two different species? What is the difference in the DNA?
It's a good question. So I think if you look at the common ancestor, that's much further in the past, usually, and definitely for humans, and so they're and Neanderthals. So they had a common ancestor that was millions of years before actually the two that the exchange that was now found apparently took place. So in all these times, gradually some modification occurs in certain sections of DNA. Certain sections of DNA are able to change to adapt to certain traits, to how species develop. Other parts of DNA are particularly stable and they don't change, or they change at much slower rates. And that's why you can have certain timers in analysis of genome in order to decide how long ago certain things occurred or were introduced in a species. Again, I'm there absolutely not an expert in how this happened, but the people have ways of defining, finding out when things happened in the past. Yeah, it's a bit like carbon dating at some point. You know certain things have happened and you can trace back because of how long ago certain things happened to the DNA.
Yes, I'll start with you, and then we have to see if there's time left till 8. Yeah.
I'm really sorry, because I have 2 questions, but also maybe that will be answered really quickly because it's really out of your field. So seeing as Neanderthals and Homo sapiens seem to have fertile offspring, otherwise you wouldn't see it in the DNA nowadays, do you know how these findings changed our perception of Neanderthals being actually a separate species from Homo sapiens? There was work published which traced mitochondrial DNA, which shows family relationships. And what that work apparently shows is that they can trace back the roots of humanity to basically 3 individuals, so 3 mothers.
Yeah, okay. Yeah. To start with the first, I think absolutely it changed our views of Neanderthals. And there have been quite a flow of papers after that coming about how this coexistence might have occurred and whether they may have even formed joint groups and stuff like that. So this whole, you may even call it misplaced arrogance of our own Homo sapiens being better immediately than Neanderthals, I think has been revised. And I think the view is now more that they really coexisted and had for a long time similar ways and similar attitudes and similar ways of living, which for a long time kept both running. And so then the question comes, why did Neanderthals in the end did not survive? That's then a different thing to investigate. Mitochondrial DNA is tricky, and that's something you can read also on the website of the Nobel thing. That there's a whole description that mitochondrial DNA, the tracing indeed only worked from mother to offspring. So you cannot trace information coming from the father that way. That can only go through the nucleus DNA. And therefore there are limitations to what you can actually do by looking at mitochondrial DNA. Yet there's a lot of this timing issue is found through mitochondrial DNA, and then the timing tells you, and combined with where you find things, tells you a lot about when certain tribes started migrating and which things occurred when and coexisted with other species at what location. And so this mapping of what species occur at what place and what time is of course what makes this whole game so important, but also extremely difficult to do correctly. And that's where this technique hopes to contribute to.
Since you are next to the microphone, if you can make your question very short and the answer as well, then we can allow it.
Two questions.
One.
One is, can you see by DNA sequences already a new kind of human arising? And the other question is, while DNA can be demolished, can you see in the DNA what kind of diseases one can expect in the near future?
The first one I find very difficult to answer, and I'm not sure that there is actually. I guess there must be people looking at that and wondering, what can you look at trends of how DNA is evolving and what would be next. Not sure. I think at the moment, my personal experience, I think, is that I think we see cultural things happening more quickly than maybe changes in the genetics. So, but we can discuss that further maybe later. The second question, you're absolutely right. You can read from DNA whether there are certain defects potentially there or risks on getting certain diseases more than others. So there's a lot known in genetics about how certain genes and certain specific sequences in those genes may signal diseases occurring later in life or when coexisting with other environmental factors and stuff like that. So there's a lot to learn. It's not always black and white, that you must get something if you have this, but there could be a higher risk for getting one disease or having, being more energetic or I don't know what. So there's definitely a relationship between genetic information, which we can now read and start to understand better, and how we behave, build, react upon our environment, and etc. Yes, absolutely, including health. Yeah.
All right, that brings us to the end of this meeting. I know there are more questions, and please feel free to contact Jurriaan. I'm sure there will be time, a few minutes for interactions further if you want to, but I want to close because some of you may have to go elsewhere this evening. It was a great pleasure to see you all here. Keep in mind that Studium Generale has a very interesting program throughout the year. It's advertised, and for those of you who are not so often here in Studium Generale, take the opportunity. It's a fantastic program with great contributions. For Jurriaan and Pepijn, please stand up, Pepijn. We'll give you one more hand of applause. And you will be forever in our famous list of Nobel Lectures for the University of Twente, so that's great. And Peter, thank you again for hosting us and all the other members of the Studium Generale team, and have a good evening.
Yes.